So hi, hello again, and today's topic, glycolysis. A super important topic, I think a very interesting topic. Not many people like it though, because it's a little bit abstract. And it's biochemistry, it's physiology, metabolism, and I think it's nice because it's very logical and very structured. But again, it's kind of difficult to imagine the processes a little bit if you're not used to, let's say, chemicals, chemical notations and so on. But let me explain it first. Well, first of all, let me give you first the bigger context rather. Glycolysis is the splitting of glucose inside our cells to release energy. So why is this even important? Well, you have to know, or you already do know, that one of our main energy sources is starch. So worldwide people eat a lot of starch: rice, potatoes, wheat, beans, corn. Okay, it doesn't matter. It's starch. And it's digested, and ultimately the starch is broken down to glucose, and the glucose transported in our blood to the body cells, and then there the body cells break down the glucose to release energy. And glycolysis is this very first step in the breakdown of glucose to release energy. And there are many enzymes involved, but I'm going to explain to you today the steps of glycolysis without using too many chemical formulas. But there is one thing that is always important, and that is, let's follow along the number of carbons. That's, I think, the most important thing here. Ask yourself, what happens to the carbon? And the second important question always ask yourself is, where does the energy go? Because as you all know, we cannot make or destroy matter. That's the carbon, and we cannot make or destroy energy. So the energy must come from somewhere and must go somewhere. And those two things, matter and energy, we're going to keep on tracing throughout the biochemical pathway. Okay, so and that is basically where I'm going to start. I'm going to start making a little diagram here on my whiteboard, not a diagram, but really the metabolic pathway, a simplified metabolic pathway of glycolysis. I'm just going to put this here, and then let's start a little bit by looking a little bit at the name itself. What does glycolysis even mean? The glyco refers to glycose, and what is glycose? Glycose is the old term for glucose. You don't have to know that, okay? So forget about it right away. But the lysis part, that's actually the more interesting one here. And that means that's a splitting, okay? Splitting. So glycolysis is the splitting of glucose. Okay, and this is actually what happens. Remember, of course, glucose must be broken down. And what happens during glycolysis is we chop the glucose in half. Well, the cells do that. The enzymes in our cells do that. And in this process, energy is released. Okay, and I'm going to show you now how these steps happen. Okay, but I do need the space, and for this reason I'm going to wipe it off again. And what I'm going to do now is I'm going to simply start off, and I'm going to start off with glucose here at the very top. So I'm going to write down glucose. Okay, and let's trace the number of carbons. We know that glucose is C6H12O6, C6H12O6. I'm not so much worried about the full formula. I'm just writing down this. It's a C6 substance. Okay, there are 6 carbons. And you know what I'm going to do? I'm going to draw all of the chemical reactions in a different color, because it's kind of nice to change colors every now and then. And then I'm going to change color again. Okay, I've over here, I don't know, let's use green, and for the labeling part. Okay, so and what happens in the first step is that glucose is phosphorylated, and to form, and I'm going to show you how to, let's write down first what we make. Hexose, supposed to be an X here, hexose biphosphate. So hexose means it's a 6-carbon sugar. It's actually fructose 1,6-bisphosphate. You don't have to know this, okay? But there is there are certain names here. But this basically means it's still a C6 substance, okay? But there are now 2 phosphates connected to it. And essentially what happens now? Well, the thing that happens is we're adding energy right now. And I'm going to, as I promised, I'm change color a little bit here. So let's do it like this. And what we're doing now is we're adding the energy of 2 ATPs molecules, ATP, and ATP is an energy carrying molecule. And what we're doing now is we're transferring two phosphates, one phosphate from each ATP to the glucose to make hexose biphosphate. And in the process we're making as a result 2 ADP. Okay, so that basically it's an adenosine diphosphate and this is adenosine triphosphate. So yeah, two phosphates have been transferred to the glucose to make this. Now why do we do that? Or why would the cell do that? What's the whole point of this? Well, this here is an energy investment stage. So we're adding energy to glucose. It's kind of strange. I thought we were supposed to get energy out, right? Isn't this the whole point? What I just told you, we want to get energy out. And what I have to do here, however, is I have to invest energy. It's a little bit like striking a match. When I want to light a match, I have to invest a little bit of energy first so that I can get more energy out later. Okay, so this here is the activation energy or energy of activation. Glucose is too stable. Glucose does not want to react, and to make glucose more reactive, I have to add energy, and this is what I'm doing in here. Okay, this first stage has a name, and this is what we call phosphorylation. Okay, let's move it over a little bit. Phosphorylation. It's the phosphorylation of glucose, and it's the first stage or the first process in glycolysis. That's energy investment. Energy investment. Okay, very critical. Okay, and this hexose biphosphate is unstable. I mean, that's the whole point why we're adding the energy here is to make it unstable. And as a consequence, the hexose biphosphate is now going to split in half. And this is now the lysis part. Okay, so we are going to make another arrow over here. Okay, and we're making a substance, two molecules now, because when we split this hexose biphosphate in part, we get two molecules. And these are known as triose phosphates. Okay, and two of them. Okay, when we split it in half, we get two parts. And this stage over here is known as lysis. And that is this lysis part up here. Yeah. So number 2, lysis. So the lysis part is now the splitting. And it is possible because we made it unstable first. So we have invested energy so far, and what we now want to do is we want to get some energy out. Okay, and I'm going to draw another arrow, and this arrow is going to be slightly longer because there are now 2 steps involved here. Okay, and again, the end product, I have to now move it up. The end product are now 2 molecules of a substance that we call pyruvate. Pyruvate, it's supposed to be a U over here, pyruvate. And this is basically 2 times a C3 substance. Okay, unfortunately, the reflection of my light on the whiteboard is a little bit disturbing. So it's 2 times a C3 substance. And of course, this is the triose phosphate is of course also 2 times a C3 substance. Okay, so maybe 2x, it doesn't matter. It's the same thing. Yeah. So what happens here now is the following. We now make 2 arrows over here. Okay. Now the first arrow over here is the production of something that we call NADH. Okay, so I'm also going to write this down now over here. What we are producing is we're producing 2 molecules of NADH, and in order to produce that, we have to put in 2 molecules of NAD+. Now what happens here? This stage is known as oxidation, and the NAD+ will remove now electrons from the triose phosphate. So this is an oxidation process, and that's why I'm going to label over here number 3. That's an oxidation where NAD+ removes electrons, high energy electrons, and they are then stored in NADH. Okay, so this is basically where energy is harvested. We're getting energy out now in the form of high energy electrons. And the second one over here, what happens here now is we're getting ATP out. And what we're doing right now is we're putting 4 ADPs, adenosine diphosphates, in, and we're getting 4 ATPs out. And that's a lot of energy. So notice we're getting out 4 ATPs, but we invested 2 at the top here. So we are gaining 2 ATPs. Okay, so this stage number 4 is known as ATP formation. Number 4, ATP formation. Okay, ATP formation is stage number 4 over here. Yeah, and collectively, both of them together, you know, I should actually make maybe some kind of brackets over here. This is our energy harvesting stage. You know what? Why not use a different color because it's so fun? That's the energy harvesting. We're getting energy out, and up here we have our energy investment. Energy investment. Okay, we have to invest a little bit of energy first to get more energy out. And as a matter of fact, we're not getting out energy only in the form of ATP, 2 more than we invested, but also in the form of high energy electrons. Yeah? And this basically shows that when we break down molecules like glucose, okay, we chop them in half. Yeah, we essentially also get the energy out that was stored in the bonds of the molecule. Okay, because of course the pyruvate that we're making down here, of course, now carries less energy than the energy of the glucose, because some of the energy is now found in NADH and also in ATP. Okay, so this is basically the whole process that we have of glycolysis, and I highly recommend that you memorize those steps here. Okay, sometimes there are multiple choice questions that ask which is the correct sequence of steps, and it's kind of obvious and very easy that you have to invest energy first to make the molecule, the glucose, unstable, and then it will fall apart and break. Break is not the correct term to use here, but enzymes have an easier time to split the molecule into 2 parts. Yeah? And then we can go further down the pathway where we get the energy out, and then we have pyruvate as the end product of glycolysis. Okay, so and what I would like to do now is I'd like to summarize this again by using again my little sheets of paper that I have. Okay, so let's put the board away, and let's put down again always include a little heading here. And when they ask you some question about what is glycolysis, then always say, well, it is when glucose is split into 2 molecules of pyruvate. Okay, this is basically what happens. We're splitting glucose, and then the glucose is not there anymore. And it occurs in the cytoplasm of the cell. And which cell? Well, pretty much all cells. Okay, it's a very, very basic biochemical pathway that you can find very, very widely in nature. And it does not require any oxygen. Why is this important? Well, this basically means that glycolysis was present already before there was oxygen on Earth. And this was actually one of the ways how very early cells were able to release energy even in the absence of oxygen. So this is actually quite important here. Yeah. So then there is an initial energy investment needed of 2 ATPs. That is our energy of activation to basically make glucose more reactive. And as a consequence of this, we are able to produce 2 molecules of ATP net gain. That's important, okay, that we're making 4, but we're gaining 2, and that is why it's called net gain. So if there is again some kind of a question here, always check whether they actually mention if it's a net gain or not. And we're also producing 2 molecules of NADH. These are carrying energy in the form of high energy electrons. So both of them here carry now the energy, or some of the energy that was stored in glucose. And of course, there's still a lot of energy stored in pyruvate, and that's why we have to, in the following steps, we have to break down pyruvate yet further so that we're also able to release the remaining energy that's still stored in pyruvate. Now, Glycolysis, as a final comment, is not only important for breaking down glucose to release energy, but because there's so many intermediate steps, it also allows the cell to convert glucose to other important substances that it needs. Okay, pyruvate is not only there to be broken down and further, but can also be converted to other substances that the cell might need. So you see that the biochemistry here is quite important for multiple reasons, both for providing cells with energy, but also for providing the cells with the necessary building blocks to make other cellular substances. Yeah, but that's, I think, a topic of a different video right now. I think I'm just going to leave it at that. I know it's a lot of information in a short time, so I recommend that you maybe watch the video again if this was a little bit too fast, and definitely practice drawing out the stages of glycolysis, because once you know that, then essentially, you're pretty much on the safe side. But for today, or for right now at least, I wish you all the best, and bye-bye. See you around next time.